উইথ কম্পিউটার সাউন্ড, ওই সাউন্ডটা দিন, তাহলে দেখা মানে শোনা যাবে আমাদের।
কি ঝামেলা রে বাবা, তো এর পরের দিন আমি গুগল মিটেই করব প্লিজ।
Yes, ma'am. Yes, ma'am.
ওটা আমার
Google Meet
সব
is
জানা,
best. Google
ঘোড়ার
Meet is
ডিম
best.
এটা আমার বিরক্ত করে।
ইয়েস ম্যাম, কোনো অসুবিধা নেই,
ঠিক আছে,
আপনি একটু লিংকটা শেয়ার করে দেবেন না
হ্যাঁ
হোয়াটসঅ্যাপ
হ্যাঁ
বা।
দাঁড়া আমাকে শিখিয়ে দে এটা কি, এটা কি করবো বল।
ওই তো যেখানে প্রেজেন্ট করছিলেন মাইক্রোফোনের পাশে, ওখানে যান, ওখানে গিয়ে ক্লিক করার পরে একটা থাকবে যে কন্টিনিউ উইথ কম্পিউটার সাউন্ড, ওই সাউন্ডটা আর ওই স্ক্রিনের সঙ্গে এই দুটো করলে তাহলে চলবে।
ওকে, এবার পাবো, দাঁড়া।
Talk about the origin of life.
না না
আরে
Yes, ma'am.
চুপ চুপ,
আস্তে আস্তে।
ইয়েস চুপ।
We know that all living organisms on Earth are made of cells, and that everything on Earth is related by some common ancestry. But where did the first cell form, and how did it happen? How do we even know that this is possible in the first place? As our understanding of chemistry and biochemistry became more and more sophisticated in the 20th century, we began to learn about the components of the cell, especially DNA, which we call the molecule of life, since it carries the genetic code for an organism. We began to understand the processes it undergoes, like replication, so that every new cell can have a copy of all the DNA, as well as transcription and translation, which dictate exactly how DNA serves as a code for all the components that make a species unique. We began to wonder, how could these molecules have assembled by themselves billions of years ago? A crucial experiment was performed by Stanley Miller and Harold Urey in the 1950s to shed some light on this mystery. They set up a system that was meant to mimic conditions early in Earth's history, just after it cooled down enough for liquid water to exist. In this apparatus, they placed water, ammonia, methane, and hydrogen, all of which was exposed to heat as well as an electric current meant to simulate lightning. After about a week, they found that the reaction mixture contained a number of different amino acids, which, as we remember from biochemistry, are the building blocks of proteins. Since proteins include enzymes, which catalyze the formation of nucleic acids like RNA and DNA, this offered good evidence that it is indeed possible that the basic materials needed for life could have been generated spontaneously. Furthermore, this experiment was only a week long, so 500 million years of molecules sloshing around in the primordial soup could conceivably produce much more impressive results. These findings prompted further speculation as to the specifics of how the first primitive cell could have formed. If small building blocks like amino acids and nucleotides came about spontaneously, how did they then polymerize to form larger molecules like proteins and nucleic acids? How did the plasma membrane of the cell come about to encapsulate certain molecules and define the dimensions of the cell? Some believe that biomolecules first arose near hydrothermal vents at the bottom of the ocean, where heat emanated from the hot core of the Earth, catalyzing chemical reactions and producing a variety of organic compounds. Others say things are more likely to have begun in mineral-rich tidal pools, where all manner of catalysis could have occurred on various solid surfaces. Some even propose that the first organic compounds were transported to the Earth from space, arriving on meteors. This is called the panspermia hypothesis. Whatever the case may be, we don't know exactly what happened. But with a firm understanding of biochemistry, it's not much of a stretch to assume that the basic organic components delivered from the Miller-Urey experiment were able to polymerize and were then serendipitously encapsulated in a lipid bilayer, which we know can form spontaneously because of hydrophilic interactions between the polar heads and water molecules and hydrophobic interactions where the nonpolar tails hide away. And there you have the first protocell. This first cell must have been incredibly primitive compared to even the simplest ones today, and subsequent cells grew in complexity over time. This was due to the ability of nucleic acids to self-replicate, as well as mutate, which is what led to the evolution of all the diverse kinds of life on Earth over the eons. We will learn more about evolution later. First, we need to learn a lot more about the components that can be found inside of different kinds of cells. So let's learn about all these components now. Thanks for watching, guys. Subscribe to my channel for more tutorials. Support me on Patreon so I can
Was it good?
Yes, ma'am.
Yes, ma'am.
Yeah.
Yes,
So,
ma'am.
okay. So, as today time has exceeded, should I continue or stop for today? Whatever you
Continue,
choose.
ma'am.
Continue. Please continue.
We have no class.
Many students are not there.
We don't have any classes now.
Okay. Let me start. If you feel bored, you please inform me. I don't have any problem to repeat any query, or I will be showing videos to make it in understanding and interesting.
Okay, ma'am.
Okay, ma'am.
Is it visible?
Yes, ma'am.
Yes, ma'am.
This is the whole syllabus. Cell biology, biochemistry, microbiology, immunology, molecular biology, and bioprocess development. So this bioprocess development, maybe another teacher will be teaching, and I will be teaching cell biology, immunology, and half microbiology. So let's start cell biology. So what is life and how it originated? Perhaps you have some idea. So there was some confusion. First the egg came or the cock came, hen came. So what is life and how in the earth where there was no existence of life, what was the first living thing came? So as you have heard in the video that the electrical lightning phenomenon, there was some experiment done, but naturally it happened. It is supposed to have this way. Nobody knows how it is done perfectly, but we suppose likely that in the earth, when in presence of lightning, at the time of lightning, the sparking and the lightning, they made the electrolysis of different gases in presence of different molecules, the generation of anions, cations, and the lysis of a large molecule into ions happened. And these ions, when they just made with each other, the hydrophilicity, these phenomenon were understood by the scientists. That is, the living thing at first didn't have any genetic material inside them. It was understood. It is understood like this. We don't have any evidence, but it is conceptualized that how first the life came. So the molecules, these are the cell, it is a single cell having just encoded, encapsulated by the membrane. It is called the plasma membrane. Can you see the cursor? Can you see the cursor?
Yes, ma'am.
Yeah.
Yes, ma'am.
Yes, ma'am.
And the molecule outside the cell, these are the molecules. What are they? Different types of gases are in the nature naturally. Methane, ammonia, hydrogen, oxygen. So at the time of some electricity. So when in nature how electrical pulses happen at the time of lightning. So in that time the lysis of a large molecule is dissociated into small molecules and the ions, and they subsequently develop by joining each other and form some living bodies, and outside the living bodies there are some molecules. Now how to see the molecule? The evolution of our visualization capacity. At first, how the microscope looked like? Leeuwenhoek, the first microscopy was discovered by Leeuwenhoek. It was like this, a simple holder over a platform, and the lens were kept here, and over a holder your sample is placed, and it can be visualized by adjusting the focusing knob. It is as simple as like this. And whenever the microscopy was developed according to the need of mankind, because the small you want to see, the resolution of the microscopy, it should be enhanced also. The Robert Hooke, they just drew the molecular organization of a cork under the microscopy. What is the cork? The cork we just cap with some cork. The bottles are capped, so they had some typical organization, their organization. So the first microscopic view was done by Robert Hooke, and he drew like this. The light microscopy was subsequently developed, and they seems to look like this. It is the eyepiece, it is the lenses, they are the lenses. There are different magnifications of the lenses, 10, 20, 40, 100. According to your need, you can adjust the lens and you can focus, and the sample is put over a slide in this black platform, and there are light source. Light comes from just down to up. Sometimes light comes from up to down. There are different types of microscopy: inverted microscopy, light microscopy, compound microscopy, electron microscopy, scanning electron microscopy, transmission electron microscopy. Several microscopic are there. Normal microscopes, their light source interact with the sample, whatever sample you have put in the base of the microscope. But in case of electron microscope, the electron beams, it comes from this passage and it interacts with the sample. And the basic fundamental theory of seeing the picture under microscope is just seeing the shadow. Means whenever light interact with a dense particle, it creates the shadow, and the picture is formed through this one. where light passes, it just gives the translucent view, and where light get hindrance, there the shadow, different types of shadow, it is analyzed by the microscopy, and accordingly the picture is seen. And in case of electron beams also, the electron dense materials, they are put with the sample, means the sample is stained with the reagent which are electron dense. So the electron dense are the negatively charged one, and the electron beam is also negatively charged one. So both the negative thing, when they interact with each other, the electron beam is dispersed, it is reflected, it won't pass, negative, negative, they will repulse each other, and it creates some shadow. So here the electron beam creates shadow, and through electron microscopy you can visualize the picture. So the light microscopic picture looks like this. This is the cytoplasm. Can you see the outer morphology of the cell? It is the nucleus. The inner roundish portion is nucleus. The innermost round, some 3D appearance, it is the nucleolus, and this spreaded portion of the cell is the cytoplasm. Cytoplasm is the main base of the cell, where the cytoplasmic semigel matter, it is filled with this one, and the cellular organelles, they float in between the cytoplasm, and inside the cell, the main genetic material, the chromosome, in the higher organism, nucleus is there, but in the lower organism, the protista, the what is told, the lower versions of the bacteria, they don't have the well organized nucleus, so they don't have well organized genetic material. But in the higher cells, there are well organized nucleus which carry the chromosomes. They are the genetic material. From one generation to another generation, the basic characteristic features of our body system, they are carried from one cell to another cell. Means when the female ovum is fertilized with the male sperm, both the genetic material from father and the mother, they comes together, and after being fertilized, the egg, this egg carries both the genetic materials of both the parents. That is why the offspring, means the baby, carry both the features of both the parents. Which feature is more dominant? Apparently, physiologically, from the outer look, we can say that you are more like of your father. You have curly hair, you have bright complexion, or you are more like your mother, that is, she is having sharp nose or tall length. So the dominant feature of the chromosome, it gives the dominance character, but it doesn't mean the recessive gene which is coming from the another parent, it is not there. All the genes are in between us. All the genes means the counterpart of the gene from both the parents. But which gene is more dominant? The expression of the characteristic. Each and individual feature is responsible from the decoding of a specific gene. Is it very tough for you? The language is very tough for you?
No, ma'am. No, ma'am. It's okay.
So the gene, each and individual gene, when they transcribe, first the step is transcription from DNA. Think simply, the chromosome is a entanglement of a long length of threads. What is the thread? The thread is the chromosome, and the chromosome is made up of DNA, nucleosomes, and other factors. So the long thread of DNA, they are so long you can't imagine. But all the length, the whole length is not the coding one. Specific, very few sections of the gene is having some meaningful coding sequences. Means if you write A, B, C, D, E, F, G, H, they won't lead to a meaningful sentence. You have to create first word. Then word after word, they will be creating some meaningful sentence. Then after putting the meaningful sentences one after another, a paragraph is developed. Putting some several paragraph one after another, you can make a essay. And by putting 2-3 chapters of essays, you can write a book. The simple thing as this as is how your body is made up. The gene, there are amino acids. Have you heard of amino acids?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
There are
Yes, ma'am.
building blocks of DNA, proteins. First think of proteins. So the amino acids are the building blocks of the proteins. They are composed of adenine, guanine, thymine, cytosine, A, T, G, C. So these, when they are put in a meaningful manner, AUG, ATT, TUG, like this, so they will be creating some codon. 3-lettered sequence is forming meaningful codon like word. When the meaningful codons are set side by side, they are creating messenger RNAs, mRNAs. And the messenger RNAs, when they are transcribed into the meaningful sequences, it is called the transcription. And when transcription is finished, after post-transcriptional modification, there are some finishing. All the time some finishing is needed. Because when we write a essay, writing the whole essay, we recheck that one and we correct, no, this letter, if we replace this letter, it will be more interesting, more aesthetic value will be put like this. Nature has also created like this. Some post-transcriptional modification happens and the finished product, they are translated into proteins. So there are translational modifications also. So from RNA to DNA, DNA to mRNA, and from mRNA to protein, it is called the central dogma. And the main building block of our body system is water and protein, then fat, then carbohydrate. There are macromolecules. And we are talking about the genetic material which are mainly composed of DNA, RNA, whatever it is. So, whenever you see, now concentrate in this picture, the light microscopic normal picture is looking like this. Whenever it is a fluorescently tagged staining is done, I won't say antibody because you don't have any conception of antibody. When I teach immunology, I will be talking about antibody. Now you just understand there are some staining materials. These staining materials, they are fluorochrome materials, mean in presence of certain wavelength of light, they absorb light in one wavelength and they emit light in another wavelength and giving some characteristic colored appearances. So, if you choose different proteins to be stained with different fluorochrome material, they will be looking like this. Just see, there are green cytoskeletal appearances, there are red or orange dots, there are purple roundish structure. So, by putting different fluorochrome staining with some antibodies, the cell is stained, and as different molecules are recognized by different stains, you can identify the expression of these protein by these red markers, these cytoskeletal molecule by this green marker, these purple nucleic acid molecule by the purple marker. So, light microscopic view of a cell, living cell, looks like this. And electron microscopic view, you can see there are black dots. It is endoplasmic reticulum. I will be saying you what is endoplasmic reticulum in the next class. Now just have a look. They are internal organelles of a cell, and these are the black dots. Can you see there are black dots? These black dots are the shadow of the electron dense material, and with this shadow, as shadow is black, these dots are looking black, and you can visualize these are different size black dots, these are different size black dots, and by identifying the measurement of the black dots, you know which organelle is giving the signature of which electron dense material, because their size is different. Now, if you look like this, the cell of some variety of shapes and sizes. Number A is the drawing of a single nerve cell from the mammalian brain. One nerve cell looks like this, so large and so surface covering, they have their length and breadth. Number B is a paramecium. It is a protozoa, a single celled organelle. The whole organism is a single celled one, having some ciliary structure over there. Number C is a chlamydomonas. It is a single cell green algae. Algae, do you know what is algae? The green color, in Bengali it is called the shaola. I don't know what is in Hindi it is called. The green color, হ্যাঁ কি বলে রে হিন্দিতে কিছু বলে জানিস? Algae, it is the English word. So algae, if it is not known, it is a green color plant organism which is a flora of a wetland. Means in wet area, in water body, they grow, they live. They have some flagella. Do you see the flagellary section? They can swim. Paramecium also can swim. They have cilia. So they are free moving. Number D is the Saccharomyces cerevisiae. It is a yeast cell. It is also a lower organism having the budding. From one yeast cell, the budding is occurred, and they can reproduce themselves by just budding from their body, and they can make themselves from 1 to 2 to 4 to 4 like this. And these yeast cell, they are used in bakery. They reproduces itself by processes called the budding. Now the fourth one, that is fifth one, E. It is the Helicobacter pylori. It is the bacterium, a causative agent of a stomach ulcer. So you can visualize there are different types of cell in the living world, and they have different functions. Now this is the last slide for today. I will be teaching how DNA is being replicated. We have told that DNA, the main composition of our genetic material, they are double stranded, and the double stranded DNA, they are linked with each other, deoxyribonucleic acid, and they have the adenine, guanine, thymine, cytosine, these different colors you are seeing, the blocks, and they are interlinked with each other. And these nucleotides, they, when they want to replicate, they have to dissociate from each other. The double stranded one, they become single stranded, they unwind themselves in presence of topoisomerases, different enzymes are there, and when they become the single stranded one DNA, the mRNA, the RNA synthesis happens. It is called the transcription, and from the transcription the protein synthesis called the translation happens. The amino acids, the building blocks of the protein, they are developed. So the sequence of nucleotides in a particular segment of a DNA, a gene, is transcribed into RNA molecule, which can then be translated into the linear sequence of amino acid of a protein. So the protein is a macromolecule, and if you visualize a sentence and it is dissociated into words, so the protein is a sentence, the amino acids are the word, and the only one small part of the gene, RNA and protein, they are shown like this. So the living organism, they can be surviving in the world as a colony of bacteria, as a small living being. It is the arthropoda, it is a butterfly, it is a plant, a rose, and it is a mammal, a whale. So penguin, whale, whatever you see in the seas. So the copying is not the perfect. Then what is copying? A cell produces by replicating its DNA and then divided into two. Means whenever a cell division, because when the first fertilized egg happens in the mother's uterus, and now a full grown person have multiple cells, millions of cells. So from a single fertilized ovum, how these numbers of cells are created, this is the cell biology we have to read. And by just duplicating the DNA content, they only can just reproduce from one cell to two. Otherwise, if we have just think in a kg manner, means you have 50 gram of DNA in the first stage, and now you have 50 kg of DNA. So from 50 gram to 50 kg, how it can happen? You have to just replicate, you have to double the DNA, and this doubling happens by DNA replication. And the passing of a copy of the genetic instruction encoded in its DNA to each of its daughter cell. From one cell to another cell division, it is happened, and the new cells are called the daughter cell. That is why the daughter cells resemble the parent cell. As the DNA is replicated, so the resemblance of the daughter cell, there are resemblance, because whenever the germ cell, that is the ovum and the sperm, there are recombination chances. But in somatic cell, whenever one fertilized egg is already done, they have to just increase in number. No need for chromosomal exchange, no need for any variation. So it is only done by the cell division, simple cell division. It is called the mitosis, and the daughter cells, they resemble the parent cell. It is not the father mother parent cell. Suppose the baby is having its small hand and now your hand is large. So how, when you were a baby, this small hand has grown into more numbers of cell. The parent cell, the baby parent cell has to be multiplied to have your adult parent hand. So these parent cells are just the copying of the parent cell. But the meiosis happens when the fertilization is needed. The parent cell of father and mother, they has to come together. The crossing over of two genetic material happens. The recombination of the characteristic feature happens, and the offspring means the baby carry both the characteristic features of the parent. But in this case, when the multiplication of the cell to just give the volume to have the growth, these parent cell, they don't need any recombination, just copying of the DNA, the replication of the DNA, it is needed, and the copying has to be perfect. Otherwise the baby hand, if it is not copying properly, the cell copying is not proper, then the adult hand will be different from its babyhood. It is not needed. However, the copying is not always perfect, and the instructions are occasionally corrupted by mutation. That is why we tell them mutation. Whenever some problem is seen, it is called the mutation that change the DNA. It changes the DNA. For this reason, the daughter cells do not always match the parent cell exactly. So, mutation, it is not a good thing at a glance. Sometimes mutation is good. We will let you know in the chapter of immunology. But mostly whenever mutation occurs, it is not good. It gives you a wrong information, some mismatching. Whenever the babies write some sentences, they wrongfully write some words. We also do spelling mistakes. So these spelling mistakes are the mutations. So the all types of cell of our body originate from the zygote. Zygote means the fertilized ovum. Whenever the fertilized ovum, they generate into the embryogenesis happens, means from a fertilized ovum the multiple cells are originated, give rise to a full grown baby. And whenever in the mother's womb, the all the organelles, all the organs, they are developed. The after the trimesters, the 9-month 10-month time is passed inside the mother's womb with full nutrition, then the baby is born. Now all the growth of the organs of the baby, they are just copying the DNA of their own body. There should not be any type of miscopying. Otherwise mutation happens. Wrong writing, spelling mistakes creates mutations, spelling mistakes in where transcription or translation. So the proteins has to be created, generated. The carbohydrates has to be generated. The fats, lipid metabolism has to be done. That is why we take food, different components of food. We take protein, carbohydrate, minerals, lipids, waters. Understood? Yeah.
Ma'am,
Just
ma'am,
a
anyone
second.
is kicking out from the meeting.
Yes, ma'am.
Huh?
Someone
Sorry for
was
interrupting.
Someone is
me.
kicking out everyone from
Someone
the
is
meeting.
Yes, ma'am.
kicking out
I've
from
been kicked
all
out
from the
6
meeting.
times.
I am me 2 times.
No, kindly don't do this. जो भी कर रहा है मत करो ऐसा.
Yes, ma'am.
How
Someone
it
who
is
is
possible?
cleaning.
How it
Ma'am,
is
it's
possible?
yes, ma'am, it's true.
Everyone has permission to
Ma'am,
remove
kindly,
any.
Yes, ma'am.
Yes,
Yes, ma'am.
Okay,
Everyone has
okay,
permission.
okay, okay, okay,
Someone is
okay,
doing, ma'am. I have been kicked
okay.
6 times. Can check.
Pull down,
ये
pull down.
पहली बार हुआ. जो भी कर रहा है भाई request कर दो मत कर.
Acha, hold on. Amita ma'am also told me that she was muted. So fortunately today I am not muted. So I will be taking out your permission for doing anything. Please
Yes,
don't
ma'am.
mind.
Continue,
So
continue.
yeah,
Continue. Stay.
no,
Yes, ma'am.
not for today. From the next day, I will be requesting Amita ma'am. She will be doing that. Nobody has the permission to just kicking out anybody or muting in anything. So the organizational capacity will be with me and Amita ma'am. Then it will be okay, I think.
Attendance, ma'am.
Yes, ma'am.
Understood what I told?
Ma'am, what about the attendance?
Attendance will be taken. What is the problem of attendance?
No, from today. Have you taken the attendance?
Please download, ma'am, attendance.
Yeah, I will be down. Don't worry about all these things. Just love the subject, everything will be okay. What is the meaning? If you don't like the subject, don't assimilate the subject and just getting attendance. I don't believe in all this trivial thing. Please stop recording. For today, it is enough. I am just stopping recording.